All right, folks, let's go ahead and get started. Your homework for the fluid and electrolyte chapter, is that due today? Tomorrow. Okay. So homework due tomorrow by midnight, don't forget. And for your partner projects, if there are a couple of you who maybe would be able to do those early, we may get finished with this reproductive system chapter a day early, and we could start those projects early. So I'll probably, because there's a lot of absent people, I'll send out a note to everybody asking for some volunteers to maybe do it on the day before we do our review, just to have a little more time to look at those. Okay, so I'll send something out about that. All right, so I introduced the anatomy of the male reproductive system last time. And in order to really understand why the testes are located where they are, you have to understand that they don't end up where they begin. Okay, so in the developing embryo, the ovaries in the female and the testes in the male actually begin in the same place, and that is in the just anterior to the posterior abdominal wall. So, this is the bottom half of a developing embryo, so 2 months development. This is not 2 months of age. This is 2 months of embryologic development, okay, so about 8 weeks of pregnancy. The testes develops here in the posterior abdominal wall. So well below where the kidneys are, but well above the rectum and the sigmoid colon here. Now, connecting this developing testis to the bottom of the scrotum is this band of connective tissue, okay, which in many ways is going to mark the journey or the path this testis is going to take before delivery, at least in most babies. Okay, so this is at 2 months. Now, between 2 months and 3 months, we don't know a great deal about. Well, we know that between 2 months and 3 months of development, the baby grows tremendously in size, but that's not anything unique to this particular month. The development of the human offspring from single cell to full-term baby is a lot faster than we often think of it as. Okay, so there are times when the developing embryo doubles in size and mass in a week, right? So the developing embryo is growing very quickly. So between this 2 and 3 month time, the baby is growing, but this connective tissue band is not. Okay, so even though it looks like this connective tissue band has shrunk, it just hasn't grown with the rest of the baby. So as the baby grows, this connection between the scrotum and this developing testis essentially pulls that testis down into the anterior part of the abdomen, so away from this posterior abdominal wall. Not because it shrunk, but because the baby grew and it didn't. Now then, between 3 months and birth, that continues to happen. The baby grows faster than this connective tissue band does. And also, in most baby boys, this connective tissue band also contracts in the last 4 weeks, or the last 4 weeks of development, and pulls the testis all the way, or the rest of the way, down into its final resting place in the testes. Now, this descent, we call this the descent of the testis, from its origin to its final anatomical location, does not always work perfectly in all baby boys. Okay, so one of the most common developmental disorders is what we call an undescended testis, or undescended testes. And what that looks like is you've got a newborn baby boy, and one or both testes are not in the scrotum. So one of the things that the pediatrician or the family practitioner does as part of that newborn physical examination is to make sure that both testes are in fact in the scrotum, because it's very, very common for one or the other not to be. Now, of course, parents immediately think that there's an absence of the testis, right? That the baby is missing a part, so to speak. It's not missing a part. That part is just not where it belongs yet. So usually it's up here in the anterior abdomen, or somewhere in what is called the inguinal canal, which I'll show you in a minute. And that's no big deal, because typically the testis is formed just fine. It just didn't finish its descent for whatever reason. Now, there's different schools of thought about what to do about it. Most of the time, pediatricians and family practitioners will wait for up to 6 months to see if the testis will eventually descend on its own. If it doesn't descend, well, then something has to be done about it. Because universally, a testis that is not in the scrotum, is up here in the abdomen somewhere, will eventually become cancerous because its environment is not what it should be for the testis to function normally. So you'll have a surgeon, usually a urologist, will go up here into the abdomen, find the testis, pull it down through the inguinal canal, and then put a couple of non-absorbable stitches in the bottom of it to bind it to the bottom of the scrotum. Because otherwise, if they just pulled it down, it would, in fact, go back up eventually. So they have to tack it down, they'll call it. And then once that has occurred, baby's fine. So in these cases, there's no impact on fertility. I mean, unless the testis didn't form properly, but that's a different issue. But typically, the testis is brought down, tacked down, baby has a little outpatient surgery, and goes home and is fine. So those of you who will take care of newborns, you will find these, because I don't remember what the prevalence is. But it's what you're going to find several of these in every 1,000 infants, okay? Which means over a career, you're going to see a number of them if you take care of newborns. Or it's oftentimes the obstetrician will do the newborn examination too, particularly in rural settings, so they'll see them too. So don't panic. Don't panic the parents, right? It's going to be fine. It's in there. It's just not in the right place. Okay. So because the testis makes this journey, the anatomy is a little more complex for the testis and the scrotum than in other places in the body. Okay, so this is the final adult development. So here's that testis down here, and the path that testis took out of the abdomen and down into the scrotum can still be seen in the anatomy. Okay, so there are two rings. There's a superficial inguinal ring here, and you can't see it, but there's a deep inguinal ring further up. That is the location where the testis passed out of the abdominal cavity and down into the scrotum. And this comes up clinically because it is through this superficial inguinal ring that many hernias occur. Okay, so it's a natural structure present so that the testis could get out of the abdominal cavity through all this tough fascia. But it makes a weak spot in the abdominal wall, particularly in men. Now, what's interesting is women also have the rings. They have a superficial inguinal ring, and they have a deep inguinal ring, and they have an inguinal canal. There's just nothing in it, right? Because there was no testis that traveled that path. So because this superficial inguinal ring here is kind of a weak spot, men can develop what's called an inguinal hernia. Okay, so a little physics of the body. When you go to lift something heavy, okay, that weight causes pressure in your abdomen, right? Because you're all one organism. So you bend over, and if you pick something up and that weight goes down your spinal column. Well, that pressure in the abdomen is usually contained by the abdominal muscles and the fascia, the thick ligamentous-like material that covers the front of the abdomen. Well, in these weak spots, the contents of the abdomen can push through those weak spots and form a kind of bulge. Now, a bulge, while painful because tissue has been torn in that, is not a big deal. The problem occurs when you have bowel or omentum from the abdominal cavity that gets pushed into that bulge, and now it bulges out and stays bulged out, right? So that hurts, and it can also cause digestive system problems because you've got a loop of bowel that's stuck in this little mushroom-shaped bulge. So you can imagine that's not going to allow things to pass through very easily. So what we do in the case of an inguinal hernia is first we try to reduce it, which means push the bulge back in. So you try to push the contents of that bulge back into the abdominal cavity. And that's a good thing, because if you can push it in, it means that there's no terrible emergency. However, particularly in these inguinal regions, it's going to keep happening. Every time that guy goes to lift something heavy, it's going to bulge out. So one of the most common surgeries is an inguinal hernia repair, in which case this inguinal ring gets a piece of mesh, plastic mesh, put over it and stitched in place so that the abdominal contents can't push out there anymore. So that's one version of a hernia. There's other kinds. You had a question?
Yeah. So it goes through the inguinal ring, right? The bulge.
Mm-hmm.
So wouldn't it make more sense that since women don't have something going through that ring, the bulge would happen more like onto the ground?
Good thinking, except it stays really small. So they have the ring, but there's no weak spot there because nothing ever passed through it. You know what I mean?
Yeah.
Because as the testis pushes through it, it stretches it out. That's why. Good question. So, you know, in the classic physical examination maneuver performed on males, which is the turn your head and cough, which many of you will have heard about or will perform, some of you, what you're feeling for is you're feeling for abdominal contents pushing down into the scrotum through this inguinal ring. In other words, you're looking for a hernia. Because sometimes they go unnoticed by the patient because they don't do a lot of heavy lifting or there's nothing that's ever been stuck in the bulge, right? So very common clinical scenario from the basic anatomy, which is the testis started up here and then came down here. So that's one aspect. So in between the deep ring and the superficial ring, we have what's called the inguinal canal, which is right here. You can palpate this in most males too. It really does feel, it sort of feels like a cord, like a round cord about a centimeter in diameter that goes up the inguinal ring. So the contents of that inguinal canal here are what are called the spermatic cords. There's one on each side. And this is all of the connections of the testis to the rest of the circulatory, nervous system, and reproductive systems. Okay, so because the testis started up here and then moved down, well, all of its connections had to go with it in order for it to stay attached to the body and functional. So even though the testis is way down here in the scrotum, its connections go up through the inguinal canal as the spermatic cords, and that includes nerves to the testes, arteries, there's two or more, the veins, which for the testis is more of a plexus or a network of veins. It's not like one vein, it's a complex network of veins. And then you have the ductus deferens, which are going to carry sperm from the epididymis up to the urethra. Okay, now, wrapped around all of those structures is a muscle called the cremaster muscle. So the cremaster muscle not only creates the outermost covering of the spermatic cord, but it is an active skeletal muscle, and when it contracts, the testes are pulled closer to the male's torso, and when it relaxes, the testes are further down into the scrotum. Okay, which raises the question of why are testes in the scrotum in the first place? Well, it turns out that sperm production works best, is most efficient, at a temperature that is many degrees below typical body temperature. So the reason the testes live in the scrotum is because of temperature. So by moving them away from the torso, they live in a much cooler place in the male anatomy than they would if they were up close here or if they were internal. So it's all about temperature. So one of the reasons that the cremaster muscle exists is to manage that temperature. So as the male gets warmer, the cremaster muscle relaxes and the testes get further away from the warm body, right? When the male gets cooler, the opposite happens. The cremaster muscle contracts and the testes are pulled closer to the warm torso to manage temperature. So it's because that lower than body temperature is optimal for sperm production. Okay. So I say up here, these structures are all testable, so let me make sure I went through them all. All right, the inguinal ligament is not part of the reproductive system, but it's an anatomic landmark. Okay, so connecting the anterior, inferior iliac spine to the pubic symphysis is this tough ligament right here. It defines the fold where the leg meets the trunk. Okay, and this is not just found in males. Females have this too, the inguinal ligament. So anatomically, it's where the trunk becomes the leg. Okay, that's the inguinal ligament. So it's a classic landmark. You can see it in everybody. Even people who are very obese usually still have a fold where the inguinal ligament is. So you can see it in everyone. Okay, so that's this piece. The superficial inguinal ring sits just superior to the medial side of the inguinal ligament, right here. Cremaster muscle around the spermatic cords. Oh, in the scrotum is another muscle called the dartos muscle. It does the same thing as the cremaster muscle, which is to elevate the testes or descend the testes in response to temperature. It just doesn't do it as well. Okay, the cremaster muscle is the major mover there. Inside that cremaster muscle, you've got the spermatic cords, which have a nerve, artery, venous plexus, and then the ductus deferens. All right, we're going to talk more about testi anatomy in a minute. Okay, the raphe is the septum, is the continuation of the septum that divides it into right and left halves. You know, it is a semi-symmetric structure. In most males, it's not symmetric. One testis will be lower than the other in most men. Okay. Onward. Okay, so we go into the scrotum and we look at the testis. Now this, I know, is sort of a weird picture, but it's a cross-section of the testis, so you can see the front and back. Okay, so this is anterior here, and this is posterior here. Okay, so this is the testis proper. Okay, here is that cremaster muscle that we saw just a minute ago. The testis is divided sort of loosely into these long wedge-like chambers. And inside those chambers are long coiled tubules that we call seminiferous tubules. Seminiferous tubules are where sperm production begins. Okay, so it is the site where sperm are formed. Now, the sperm that come out of these seminiferous tubules are not fertile yet, but that is where sperm are produced, is in the seminiferous tubules. Okay, so each of these wedges, because I can't remember if they call them lobules or something else, but anyway, they're wedges. I'm not going to ask you. So seminiferous tubules, there's 2 or 3 in each one. They all connect to this maze-like structure right here called the rete testis. The rete testis connects the seminiferous tubules with the epididymis in the back. Rete actually means maze or labyrinth. So it's this sort of weird structure where these different tubes interconnect. But in the end, we've got 3 ducts called efferent ductules that go out into the epididymis. Now, the epididymis is one very, very long, very, very coiled tube. So it's just one tube. So each of these are 2 or 3 tubes all coiled up. This is one tube all coiled up. And the epididymis collects sperm that have been made, and they are stored in the epididymis. They are also, they mature in the epididymis so that they are capable of fertilizing an egg once the seminal secretions have been added. And then leaving the ductus deferens, or leaving the epididymis, is the ductus deferens right here, which are going to go up into the body. Okay. All right, so we have to talk a little bit about like cell biology stuff in order to understand sperm production. So typically, what you learn about in biology is what's called mitosis. That's cell division. Okay, that's where you have a mother cell that divides into 2 daughter cells, but they all have the same amount of genetic material. They all have the same genes, right? Well, meiosis, notice the absence of the T. meiosis, or meiosis, some people will call it, is the special kind of cell division that results in gametes. Gametes are also called germ cells, and in the human, that is the eggs, the ova, and the sperm. Okay, so those are the gametes. And what makes them different than other cells is because of this different division process, they end up with unpaired chromosomes in the end. So you remember that in the human, there's 23 pairs of chromosomes. Well, in the gamete, we have just one of each chromosome instead. And the chromosomes, which are normally duplicated, are in half. So we have half the genetic material that we normally have in a typical cell. So gametes have 23 chromosomes as opposed to 23 pairs. So we call them haploid. A haploid cell has half the genetic material. Some of you are nodding very easily. You've had genetics, right? So did you talk about this in genetics? Good. See, that's awesome. So there's less to learn here. All right. So as you know, the difference is how many cells you end up with in the end and what the genetic material is like. So instead of getting 2 daughter cells, we get 4 in the end, and each of those 4 have half the genetic material. Okay. All right. So the process of meiosis in the male and female are very different. The male and female have different reproductive strategies at the gamete level. The male produces huge numbers of gametes all throughout life. The female produces a small number of gametes, 1 or 2, usually 1, at a time, once a month over the childbearing years. So we've got 2 different strategies, and we see that in the gametogenesis, or the production of these gametes. Okay, so spermatogenesis is the production of sperm cells. Spermiogenesis is turning those cells into functional, fertile sperm. Okay, so they're 2 different words. Spermatogenesis is this part. Spermiogenesis is this last step right here. Okay. So it all starts with this guy, the spermatogonium. This is a stem cell process, which means it can go on forever. Because this cell will divide, right? One of them will head down this path. The other will take the original's place. So this can go on and on and on, millions and millions of times, hundreds of billions over a lifetime. All right. So the spermatogonium becomes a primary spermatocyte. It divides into secondary spermatocytes. That's meiosis 1, for those of you cell biologists, which is not me. Those secondary spermatocytes divide again. Now there's 4 of them. Note that they're all the same. When we get to the female, that's not true. In the female, it's all asymmetric. One cell keeps getting all the resources, and its daughter cells get nothing and go on and die. So in spermatogenesis, it's all symmetric. So the secondary spermatocytes become spermatids. Spermatids then get remodeled into the spermatozoa that you've all seen in pictures and classes before. Okay? But in the end, these guys all have half, all of these here, have half the genetic material of this one. All right. So what we end up with is the spermatozoon is the singular, spermatozoa is the plural. Okay, so we get some weird Greek in there, so it's a spermatozoon. And essentially this cell, because it's not an organism proper, because it can't do all the things an organism can do, but it is a cell, and it evolved for one purpose, and that's to deliver its genetic payload, which is found here in the head, to an ova, to an egg cell. All right, so its whole structure is based on that. So we'll start at the top. The spermatozoon has a cap of enzymes called the acrosome, or the acrosomal cap. Those enzymes will digest the ova's outer protective covering, which we'll talk more about later, allowing, usually anyway, just one spermatozoon to fertilize that egg. So the acrosome has digestive enzymes to eat that outer covering. The head, which is just under the cap, the head contains the genetic material. So that's that haploid DNA, half, 23 chromosomes that dad is going to contribute to a possible offspring. In the neck, we have in the neck essentially connects the head to the middle piece, so it doesn't have a specific function. The neck, though, is kind of the engine of the spermatozoon. The neck is full of mitochondria, and those mitochondria are going to produce ATP to make this tail, or flagellum, do its thing, which is kind of a twisting and rotating motion at the same time to propel this spermatozoon in a relatively straight line, okay? So, very lean. You don't see much in the way of extra material in this cell. And one of the things you definitely don't see is there's no fuel on board. Okay, so we've got mitochondria to produce ATP, but they don't have anything to metabolize. And that is why the spermatozoa has to have the seminal fluid in order to function, because it does not have anything to burn. It's got no fuel. So one of the secretions of the seminal gland is fructose, a sugar which these mitochondria are going to use to create ATP to get this tail doing its thing. Okay? So it has to get its nutrients from the outside. All right. So by what process do we arrive at that mature spermatozoon ready to swim and be fertile? Well, the process, like I said, starts in the seminiferous tubules in here. Each of these wedges is actually 2 or 3 very long, very coiled up tubes. So when we look at it under the microscope, we see these circles. So these are circles, they're seminiferous tubules in cross-section. Okay. Now, sperm production begins around the outside. So the spermatogonia live just inside the wall here. And as the spermatids, or as the cells divide, they move in closer to the middle. Until eventually what you have stuck around the wall here are relatively fully formed spermatozoa with all their tails sticking in towards the center. You can really see that here. So like these are mature spermatozoa, and these are their tails sticking in towards the center of the tube. Now, when they're ready, they release from the tube wall, and they travel through the seminiferous tubules, through this long system, into that rete testi, and then eventually into the epididymis. All right? Okay, so they work their way in. I think I have a better picture. Yes, I do. Good. Okay. So these are the stem cells, spermatogonia, around the outside. These are the different stages, right, as meiosis occurs. And then finally, here are this mature spermatozoa with their tails sticking in towards the lumen. Now, these sperm production, these dividing sperm cells need a lot of help to finish the job. Okay, so surrounding these developing spermatozoa in purple here, this is another type of cell called nurse cells. They're still called that. They used to be called Sartoli cells. And one of the illusions in a picture like this is it looks like these blue cells are inside the purple cells. They're not. The purple cells are wrapped all around the blue cells. Okay, so they're not inside the cell membrane. This nurse cell is literally like the blob is wrapped around all of these developing sperm cells. And, doing what? Well, providing them the nutrients they need, helping these spermatozoa to attain their final shape. It's kind of unusual in the body. Usually a cell develops itself, right, from the inside using its own DNA. In sperm production, the sperm are also constructed from the nurse cells that are surrounding it, too. So it's sort of 2 cells working on the final product. Okay, so by the time they get out here, they're just about ready, and then they're released into the lumen, and they head out. So all of them, I'm looking at this anatomy to see how much of this you really need to know. Not much. I'm interested in you knowing that spermatogonia are on the outside, and then in those, the spermatids end up here, and then just inside the wall, we end up with the spermatozoa that are going to be released. And then in purple, we have the very important nerve cells that are helping to make this whole process happen. Okay, so as I said, the spermatozoa, once formed, it looks right, but under the microscope, you can't really tell the difference between a fertile spermatozoon and a well-formed one when they're stationary. So in order for sperm to be fertile, they need a couple of different things. One is what's called capacitation, or capacitation, which is essentially improving the capacity of the sperm to be fertile. And this capacitation occurs in the epididymis. So the sperm cells are produced in the seminiferous tubules. They're stored in the epididymis. During that storage time, they undergo this capacitation, where they're able to become motile and they're able to become fertile. Now, sperm at rest in the epididymis, their tails are not moving. So it's like they're dormant. Okay, that flagella and the swimming motion isn't triggered until these other secretions are added during ejaculation. So the sperm don't come on until it's ready, until they're ready to do their thing. Okay? So in order to become motile, in order to activate and start to swim, they need a couple of things. One is a fuel source. Okay, so the seminal glands, which sit right here. Okay, let's look at this picture for a minute. This, believe it or not, is an anatomical picture, but it's taken from a really weird view because it's like angled up. So you're never going to see this, but it does show you all the parts of the male reproductive ducts. Okay, so here's the ductus deferens coming in from the testes. Note they go above the ureters. So they go way up over the top of the bladder, and then they go down and under. As they join with the seminal glands, which are here and here, they form what is called the ejaculatory duct, which is what enters the prostate gland and joins with the prostatic urethra, or the part of the urethra that passes through the prostate. Okay? And then it's through the pelvic wall. So you've seen all these structures before, but not oriented like this. Okay, so from those seminal glands, we get a fructose-containing solution. This is fuel to burn, right? Because, as you know now, carbohydrates are very rapidly metabolized. So having that fructose around means that the mitochondria in that mid-piece can generate ATP. That gets the tail moving, okay? Then secretions from the prostate gland and the bulbourethral glands, they contribute additional materials which improve fertility. Okay, so for example, the prostate gland secretions are alkaline because the environment of the vagina is very acidic. So you combine those two and you get a neutralization. That allows the sperm to be more fertile. Otherwise, that acidic environment would slow them down or even destroy them. Okay, so the prostate gland adds that. And then the bulbourethral glands, as well as the prostate gland, add fluid and lubrication so that the sperm have an easier time slipping through all the nooks and crannies, so to speak, to improve their fertilization or their fertility. All right. And then we connect to the urethra. So here is prostate gland would be here. So the sperm together with the seminal fluids are coming out through here. Essentially, the penis is 3 cylinders of what's called erectile tissue. So the best way to think of erectile tissue is it's like a sponge in a tough covering. Okay, and you don't see that kind of thing very often, but it would work in the same way. So when these spongy tissues fill with blood, they have this tough outer covering which prevents them from expanding past a certain point. So what develops in these 3 cylinders is hydrostatic pressure. Okay, and that hydrostatic pressure is what changes the shape of the penis, right? So we have 2 at the top, and then we have 1 surrounding the urethra at the bottom. So the 2 large ones, these are the corpus. Each one is called a corpus cavernosum. So together they are corpora cavernosa. That's this one and this one. The corpus spongiosum surrounds the urethra, which travels along the base of the penis right here. So we have a large artery in the center of each of these, and then essentially the corpora cavernosa are veins. They're just spongy veins that normally in the non-erect penis drain the blood from this artery, but by changing the valve, so to speak, you can increase and create hydrostatic pressure. And a point I like to make is, from sheer observation, it is easy to see the external parts of the penis, but there is actually a root of the penis which goes underneath the the pubic symphysis and alongside the pelvic bones on both sides. Let me show you a better picture of that. I think I have one. Apparently I don't have one. Do I have one coming? No. Sorry about that. I've had a bunch of different slide sets for reproductive system. But you can see it here. What this doesn't do justice is it goes very much laterally as well. People think of the penis as a midline structure, but the root of it actually goes to the left and right pretty significantly. It's just underneath the pelvic bones that come down the front. All right. All anatomy. You should know, but most of this you already do know. So the two cruces, there's a right and left cruce. That's where it goes left and right underneath the skin. All right. So another weird view of the male reproductive system. Not weird because the anatomy isn't right. Weird because it's a view that's sort of down at an angle. So it's not like you usually see. But I put it in here because all the parts are here. Right? So you've got the testis down here. Here's the epididymis that sits along the back. Okay? The epididymis connects to the ductus deferens, which go up, back, around, and down. The ductus deferens have a little expansion at their ends, called the ampulla. The seminal glands join in. This is the prostatic urethra. Here's the prostate gland. Here's the bulbourethral glands that provide a little lubrication. And then you have the urethra, which travels along the base of the penis. You've got the corpus spongiosum, corpus cavernosum, and I think that's all the things we talked about. All right. So questions about any of that anatomy before we start talking a little bit about phys? Yes?
Is the only reason they're two separate organs, like the testes and penis, is it because they have to be lowered to stay out of the internal temperature? Or why is it?
Why not have them be one organ?
Yeah.
I don't have a good answer to that. Remember that before the testis is a much older organ than the penis is. So the formation of the sperm, that goes all the way back pre-fish. So there was no need to have a delivery organ. So they probably evolved separately because one was already present for 1 billion years. And then as animals put their reproductive tracts on the inside instead of doing it all on the outside like the fish do, then you had to there had to be a mechanism for delivery. But good question. See, you're thinking about redesigning a human being too.
Yeah, it's interesting how everything
they're
develops.
separate.
Yeah.
I think it's because they happened at different places in time.
Okay.
Other questions? All right. So the male reproductive tract is harder anatomically, I think, than the female's is. You can argue with me later about that. But physiologically, it's way easier. Okay, the female reproductive physiology, you're either going to love it because you're all females, or you're going to hate it because it's really complicated. But we'll get there. The male reproductive hormone and physiology system is far, far simpler because essentially, at least as far as we know, it's doing the same thing all the time. There is no 28-day cycle to it. It's just constant. Now, some research has suggested that perhaps the male reproductive system has a cycle too, but we don't really know if that's true. It certainly is not a 28-day cycle, but it may be a daily cycle, it may be a weekly cycle, monthly cycle, even yearly cycle. So Still things are being discovered about the reproductive system, in part because it just gets a lot of interest from people. So people keep looking at it. So the basics are the reproductive system is like the female system, controlled by hormones. The hypothalamus is the master of reproductive hormones and therefore reproductive physiology. Not surprising, as we learned in our first chapter together this semester, the hypothalamus controls all of the hormonal systems, right, by controlling those releasing hormones. In this case, it's gonadotropin-releasing hormone, GnRH. Let's look at that word. Gonado, that's gonad. Tropin is to activate. Okay, so this is a releasing hormone to activate the gonads. So gonadotropin-releasing hormone. Under its effect, the pituitary gland secretes two hormones that affect the male reproductive system: luteinizing hormone and follicle-stimulating hormone. Now, these hormones were named for their effects in the female, as we'll see. They are the same hormones, same chemical structure, but their names don't make any sense in the male. They make perfect sense in the female, which we'll get to. So you just kind of have to connect the two. But luteinizing hormone stimulates the interstitial cells of the testis. The interstitial cells, which I neglected to mention. Okay, seminiferous tubules, right, where sperm are produced. In between them, these guys and these cells in these regions, we call those interstitial cells because they're between. They're the cells that produce testosterone. Okay, they're also called Leydig cells, but their modern name is interstitial cells. So the testis is the principal source of testosterone in the male. Okay, and that testosterone comes from these cells right here. And that testosterone production is stimulated by luteinizing hormone. Now, why does testosterone matter? Well, testosterone is necessary for sperm to make the final transition from spermatid, little round thing, to spermatozoon, sperm-looking thing, ready to swim and deliver its genetic payload, right? So that's its critical function. Without that, the male is infertile. Okay, so you got to have testosterone to have fertility. Even though sperm production occurs, the sperm can't do anything. They're infertile. All right? So that's the principal effect, is testosterone stimulates the nerve cells, which then turn those spermatids, little round things, into the spermatozoa. Testosterone has a bunch of other effects, though, too. It affects the central nervous system and the limbic system to create sex drive and other behaviors. Testosterone has direct effects on muscle and bone. So you can give testosterone to a female, and she will develop heavier bones and more muscle mass, similar to what a male does under that influence of testosterone. Testosterone causes the male's secondary sexual characteristics. So this is things like fat distribution, body hair distribution, vocal tone, hair patterns, as many of your fathers probably know, start to lose some of the hair at the top. That's testosterone that does that. As well as maintaining the different parts of the male reproductive tract. So in addition to the infertility that comes about from no spermatozoa formation, testosterone, without testosterone, like in a patient who's had testes removed for some reason, the accessory glands and organs start to atrophy and not function. So you have that added effect, which is why typically, if a male has had both testes removed, they're on testosterone replacement therapy so that these effects can be maintained and their life can be as normal as possible. The other hormone that plays a role is FSH, follicle-stimulating hormone. In the female, follicle-stimulating hormone is what triggers egg formation. In the male, FSH is what triggers spermatid formation. Okay, remember the spermatids are this layer right here. Okay, so from here to here is all FSH driven. From here to here is testosterone driven. And the testosterone is driven by luteinizing hormone. Right? See that connection? Okay. Now, even though we have all of these inhibitions and stimulations here, their roles are not as important because, as far as we know, this system just kind of maintains a steady state of sperm production. Now, there is a kind of off switch called inhibin, and inhibin works to turn down follicle-stimulating hormone, which reduces spermatid production, which, because you have to have spermatids to have spermatozoa, ultimately controls the level of sperm production in an otherwise healthy male. This is currently a target for male birth control, because if you could work with this inhibin system to block the formation of spermatids, there would be no sperm production. No sperm production, no fertility. Now, one of the reasons why not very much breakthrough has happened in male birth control from a hormonal standpoint is because the system evolved to produce hundreds of millions of sperm every day, which it only takes one to create a pregnancy. So the problem with hormonal control in the male is it has to be complete. You know, you have to totally not have any, and the body wants to make millions. Very different than the female, where you only have to block the release of one egg at one specific time in the menstrual cycle. So, but people are working on it, developing male hormonal birth control. But until then, it's still all barrier methods. Okay, and you can kind of ignore these two boxes because they're not that important. So this, as you'll see when we get to the female version, this is simple. There's no moving parts, there's no time, there's no three-organ system like we'll see later. Okay, so we'll just got a couple of questions. Once again, we'll just do it old-fashioned way. Spermatogenesis occurs where? B, right. That should roll off the top. Seminiferous tubules, that's where sperm production begins. The vas deferens, that's the old name for the ductus deferens. If you've heard of a vasectomy, that's from that old term. In a vasectomy, the ductus deferens are clipped or cut, usually right about here because this is a really accessible spot. So they either put a plastic or metal clip there. That's a reversible vasectomy, which are not always reversible, okay, because they can scar down. Or they'll cut this and they'll tie both ends. Oftentimes they'll cut a little piece out too, because just like other parts of the body, ductus deferens do occasionally heal after being cut, and not all vasectomies work. There are lots of babies who've been born after a supposed vasectomy because these things do heal. So just so you know, anybody a post-vasectomy baby? I'm telling you, they're a lot more common than you think. It's like 1 in 100, 1 in 200. So one of your friends is probably a post-vasectomy. Okay, the spermatic cord, what is that? Yeah, I like D. What do you guys like? Okay, so let's look at these. Dense layer of connective tissue that surrounds the testis. That's the tunica albuginea, which I didn't talk about, but it was probably in the picture. The layer of smooth muscle in the skin of the scrotal sac, that's the dartos muscle. The narrow opening that links the scrotal chamber with the peritoneal cavity, that's the inguinal canal, right? And there's a superficial inguinal ring up there. And the external marking of the boundary between the two chambers, the left and right, that's the raphe. All right, and I think we're probably out of time. We are out of time. So we'll pick up there next time, and we'll get into the more complicated female.